At this point, we're going to shift to our second panel.
Okay, good morning, everyone. Again, so thank you for inviting us, and this is very much English discussion so far. I'm learning a lot. My name is Wondwossen Gebreyes from Ohio State University, Global One Health Initiative from Columbus, Ohio. We are three of us here. My colleagues, Dr. Amy Oppenheim, she's our operations director way back there, she's getting more coffee. And Dr. Girish Rajeshwara, who is also Campylobacter expert. So again, thank you. It's great to be in Gainesville, especially much warmer, maybe 20 degrees warmer. So we really appreciate the timing as well. So as Sarah and Dari asked me, I'll give a quick overview of molecular epidemiology work in Eastern Africa from the point of what we do in our partnership with the Global One Health Initiative. I'm skipping this slide. So this is really a relatively, a small part of the Global One Health Initiative. We have a broad areas of work in Ethiopia, Kenya, and Tanzania. By the way, I'm originally from Ethiopia, I'm molecular epidemiologist, but with veterinary background. Did my veterinary practice mostly in pastoral areas a long time ago. So now we work in 3 countries, but I will describe that. So food, water, vector-borne pathogens remain major issues. We started our work between Ohio State and Eastern Africa, initially Kenya, then Ethiopia and Tanzania, mainly focusing on enteric bacterial diseases and antimicrobial resistance. Salmonella and Campylobacter remain a big priorities because of various reasons, and really nutrition and the pathogen interfaces absolutely, I think, is very clear. I don't need to describe it to you much. And Sarah showed it very nicely. The impact of disease is very important, and as it's been said, can use everyone. So one of the key areas I want to focus today is what has been a progressing, expanding program that we have in the 3 countries. So Fogarty International Center D43 program that started in 2010, recently renewed for 2022. This is a busy schematic diagram, but quickly to show you, we work on foodborne pathogens, again, viral, some parasitic, but bacterial is a big emphasis. We work on some waterborne diseases, some of which also cross-interface, and others. We work on vector-borne diseases. And 3 main emphases. This is a research training capacity building program, as Fogarty is geared for, is on antimicrobial resistance is a big emphasis, perhaps the biggest emphasis. Rapid diagnostic systems, field deployment is the other emphasis, and genomics and bioinformatics is the 3rd emphasis. So any combination, could be a waterborne disease or antimicrobial or others. So you can tell I'm going to give a much broader, it's not my research presentation, but I just want to share with you further, more focusing on the enteric aspects and what our great scientists across the 3 countries are doing. And later on, I'll focus on Campylobacter, which is more relevant to cage projects. Just one slide to show you between Ethiopia, Kenya, and Tanzania, a number of fellows that were trained, about maybe 11 of them completed their PhD in the last 6 years. I've been working on very important viral disease which may contribute to ED, and as well as some other parasitic diseases, rotavirus, norovirus, other enteric virus has been a big emphasis, especially in Kenya, in all 3 countries, but especially of a number of work published through that from the University of Nairobi. Also partly vector-borne diseases were expanding now, but like leishmaniasis is a big issue on the human-vector interface that you see. Dr. Nigatu in the bottom right corner, which works with the Microsoft Systems Innovation Lab now in expanding his research portfolio. So let me quickly show you my areas has been mostly in Salmonella, but I'll give you some from Salmonella work that we do then focus on Campylobacter. Again, multi-drug resistant MDR Salmonella has been a big issue globally, but especially in sub-Saharan Africa because of this MDR, typical enteric infections, and also to be invasive systemic infections. And they're causing lots of systemic infections, acting like Salmonella typhi or cholera others that we rarely see in the US, but has been a big issue. One of our fellows, you see here, Tadesse Goala from Ethiopia, has been working on this, on several projects and several presentations came out of it. But one, you can see, for example, ciprofloxacin resistance, we rarely see in most industrialized world. We see about 15% of Salmonella isolates in Ethiopia. Depending on the studies, sometimes this is the average, essentially. What does that mean? And of course, these are foodborne pathogens, and animal source food are important carriers of drug-resistant organisms, and human beings, children, as well as adults consume. Particularly, disproportionately, children are affected through the foodborne pathogen bacteria, as you know. So getting infected through these organs through food, and then not being able to be treated is a very important global issue currently. So antimicrobial is one big focus. Number of outcomes, outputs, publications came out of Dr. Tadesse Goala's work. One of them is indicated here, but I'm not going to go much more in detail. Another Salmonella working is in Tanzania with Sokoine University of Agriculture. Julius Medardus was one of my first initial PhD fellows through this NIH program, which has, in fact, important nutrition components from the animal nutrition point of view, but important, a couple of data sets I will show you. But this is looking at Tanzania and various agroecosystems, Morogoro, Iringa, Arusha, various from pastoral and smallholder farmers. He did work also in Ohio as part of the training program, but his main interest and our interest is heavy metal HMUs, heavy metal micronutrients and their interaction. Zinc has been just mentioned as a cofactor, zinc, copper, other uses, or through the soil exposure and its interaction with antimicrobial resistance and how that translates to human health in children as well as the animal-human interface. Here is a quick slide from Julius Medardus' work looking at zinc tolerance and copper tolerance and how that interacts with antibiotic resistance. Because this is a big issue. This particular issue is a major issue for us in the US or Europe, wherever you might be. Because of zinc and copper use in animals, whether it's intensive farms or through the environmental exposure, what we notice is even if food animal production systems or withdrawing the use of antibiotics, with tetracycline or others, as long as there are zinc tolerance and copper tolerance genes co-located in various number of work starting from my own PhD, 20 years ago, as long as they are co-located genetically, they select for antibiotics. They remain to be multi-drug resistant for a number of years, for decades. And we have shown that in a number of studies, and we are now doing this beyond US, in Eastern Africa. And not only just they co-select antibiotic resistant, but depending on the copper, zinc, or whichever they use, different types of antibiotic resistance are selected. Okay, this is a bit outside of what we've been discussing, but important, relationship in terms of micronutrients. I don't know what that means in terms of EAD and others, but I think this opens interesting work. So Julius now, is a former fellow, and he's a PI in a couple of grants in his institute at Sokoine. Is expanding lots of work on that heavy metal micronutrient and also disinfectant and interactions with antibiotics. This particular, just to wrap up the Salmonella part, we have now a much broader expanded global partnership focusing on multi-drug resistant Salmonella in partnership with FDA through the Genome Tracker partnership. And the number of countries you see here, we work on Salmonella in these countries, but what is the global dynamics in terms of the various genotypes circulating in Africa, in the US, Mexico, Brazil, Thailand? We have a fairly larger project. We are now, in fact, extending this partnership to Campylobacter because of this particular caged project that Ari is leading. Quickly, to talk about Campylobacter, is more relevant to the caged project, is one of our fellows, Isaac Kashoma. You see his picture here. His primary advisor is here with us, Risha Ishara. This is another thing we extended to East Africa, is Campylobacter, much more understudied. Even in the US, it's understudied, but more so in sub-Saharan Africa. In the US also, you don't hear it. It's not a popular organism. It's a sporadic cases, but it's estimated based on CDC, estimated number of human cases in the US surpasses Salmonella. Campylobacter has a larger number, but you don't hear outbreaks, so it's not on NPR or CNN, so not many people know about it. And a very tricky organism in terms of culturing others in sub-Saharan Africa, being a microaerophilic thermophilic organism, but it's been of interest to us. So Dr. Kashoma, who is now again a former fellow and establishing his laboratory at Sokoine, has been doing a number of work. This is quickly from his work in milk and carcass swabs. He's done quite a bit and published lots of work in these three agricultural ecosystems, similar to Julius Medar's study in Morogoro, Iringa, Arusha areas of Tanzania, which we are now expanding into Ethiopia, working with poultry are very important, chicken are important reservoirs, much more important reservoirs of Campylobacter. So we're really excited about this project. And also for us, the transboundary nature of these organisms, creating the research and training network within East Africa is big interest for us as well. From Kashoma's work here, you see antimicrobial resistance proportions from Tanzania. You can see between Campi jejuni and coli, lots of multi-drug resistance. I'm not going to go too much into detail, but whether it is ciprofloxacin resistance or macrolide erythromycin resistance, you see much higher in both species, and nalidixic acid, a number of others as well. This is again of interest to us in the future as we expand Campylobacter work. Something important for us to consider is the antimicrobial resistance aspect of Campylobacter, and perhaps the chronic inflammation, what does its role in EED would be, I think, of great interest. Just to show you quickly, Kashoma did a number of antimicrobial resistance genes, exploring genes, sent or other fellows working on antimicrobial resistance to that. He identified a number of genes that are prevalent in the region, but not necessarily to go down further. But from US, in my lab itself, we have more than 3,000 isolates. We have then collected through this network in East Africa a large number. Our goal is to genotype them further with whole genome sequencing. So far, most of our work, both in Ohio, North Carolina, I used to work in, when I was at NC State, we have a number of studies in Campylobacter, has mainly the genotyping system we use so far is multi-locus sequence typing. Not based on whole genome, but based on 7 housekeeping genes, an established method for genotyping of Campylobacter. This is a work again by Dr. Kashoma, Gebreyes, and a number of people from Tanzania. You can see, again, not going to detail, but any molecular epidemiologist without showing their phylogenetic, they're not going to get that way, right? So, but not going to detail, but you see clusters of these various sequence types, genotypes of Campi coli as well as Campi jejuni. Whether it's cattle with Morogoro MR or dairy cattle in Arusha, you see we see geographic and host, clustering of genotypes. With my 20 minutes, I'm not going to much more into detail, but a number of publications came out of Kashoma's work. You can talk to Dr. Girish Rajeshkara, but definitely we are doing the knowledge transfer between Tanzania and Ethiopia. Kashoma will be co-mentor for future fellow from Haramaya that will be coming up very soon. So this being my kind of quick, overview of molecular AP work, I'm happy to discuss with anyone with more detail. But in few slides, I just want to introduce our Ohio State Global Wonders Initiative, as long as I have the platform in 12 minutes. We have, as you know, One Health is very important. What you do is One Health. The concept can be interpreted in different ways, but there are huge global risks that we all know, with a population of 7 billion of us heading to 10 billion in a few decades. With the Global Risk Forum, there are a number of areas. This is a broader view of things, and nutrition is absolutely a critical part of this. Climate change, everything is interconnected, right? The water crisis, a number of trends. And this schematics you see is from a Global Risk Forum Davos meeting that we had in 2015, I think. That shows the human-animal environment interconnection, but not only just that human-animal environment, but the peripheral urbanization, natural forest, and the broader areas. So our initiative focuses on broader areas, including nutrition, disease, and others. So our High-Stakes Global Wonders Initiative mission is really our focus global capacity building and strategic areas. Such partnership really and research helps also build capacity of our partners working in East Africa, the 3 countries, but also in other developing countries. We have our Ethiopia regional office, this is quickly our central office team, and manage our operations, and a number of 30 different disciplines are involved. And what you see on the bottom right-hand side is our Ethiopia office team. A couple of them are from Ohio State, and the rest are our regional office. We have a number of other projects on tuberculosis, global health security implementation, on rabies, brucellosis, a number of others that I cannot touch today. And we have a broader global one, it's knowledge sharing platform, what we call ICOFEL, that we established in 2011, that includes East Africa, Latin America, Brazil is a key partner, Asia, and I really want to invite you with University of Florida, a number of key areas with this emerging partnership institute for sustainable food systems, number of interfaces. So really, we look forward for strengthening partnership. So thank you so much.
Oh, thank you very much. It's really nice to be here and to be involved in this project as it starts. I'm going to tell a story about understanding the etiology of diarrhea in children in developing countries and how that's been extended to understand the relationship between enteric pathogens and child growth shortfalls. So this is what I'm going to try to tell you over the next 20 minutes. First, that we have used molecular diagnostics to really much better understand the etiology and burden of childhood diarrhea in low resource settings, and that we've started to apply these diagnostics to longitudinal studies such that we can look at the impact of these infections on child growth. And really most clearly and consistently in the studies that have been done, the pathogens implicated are bacterial enteropathogens, and in particular the ones that are invasive and associated with local and systemic inflammation. I'll talk a little bit about some of the studies that have been mentioned, the interventional studies that Mark mentioned, but studies where potentially using these diagnostics can help us understand better the success and failures of those interventions. So the standard workup for understanding the etiology of diarrhea is expensive, it's complicated, and it's very variably sensitive. So you might take a stool sample. This is from this is the schema used in a large multi-center gold standard etiologic study of diarrhea that was done started about 10 years ago, called the GEM study. So you take a stool sample, you do bacteriology. If you're getting the sample in the field, you have to add in the detail that you need to get it into transport media as quickly as possible, or you lose isolation of some organisms, in particular Shigella. For this bacteriology in and of itself is very diverse. So for Campylobacter, you use very selective media and microaerophilic and thermophilic conditions. So you're a farmer, you're trying to grow these bacteria, and you're doing it in the setting of all of the bacteria that is normally expected to be in the gut. E. coli is normally in the gut, but there are types of E. coli that cause diarrhea. So once you've grown those E. coli, you then need to look for particular toxins, particular genes that are elucidated by those particular E. coli. So you have to decide how many colonies of E. coli you want to pick to look for those. Then you might take some of the stool and do immunoassays. And these immunoassays are for targets that are specific to each pathogen and are variably sensitive. Some are quite good and some are not so good. But some organisms, for example, norovirus, unless you're going to do electron microscopy, you need to do PCR. That's really the only way to identify it. So the question comes up when you do an etiologic study of diarrhea and you find that, say, norovirus is the most common cause of diarrhea, is it? Or are you just better at detecting norovirus than you are some of these bacteria, some of these protozoa? So there's been a very unlevel diagnostic playing field for doing etiologic studies of diarrhea for a long time. This was recognized around the time that this GEM study was being done, and other studies in enterics were being started by the Gates Foundation. And my mentor at the University of Virginia was tasked with developing better diagnostics for enterics. And this is what's really we've settled on as our favorite platform. This is a TACMan array card. So what you see across the top are 8 wells, and you can extract nucleic acid, total nucleic acid, RNA and DNA, and put it into that well. And then you can just spin the card, physically spin the card, and through microfluidics, the nucleic acid and reagents that you also put in for polymerase chain reaction are spread across all of these 48 subwells. And there are primers and probes for particular pathogens or typing assays or whatever you want to include weighting there. And then you can do simultaneous quantitative PCR, real-time PCR for all these enteropathogens in one go. So when you're doing a study, when you really need to look across a wide array of pathogens, this is a really nice way to go. And it's very compartmentalized. So between labs, we've shown that we get very similar results. If you compare this standard workup to these TACMan array cards, you'll see that PCR, that the original diagnostics are specific. So when you detect an organism by, on the left, an enzyme immunoassay or culture or other methods, it tends to be detected by PCR. But many of the detections by PCR are not detected by these original methods. And there's variable sensitivity between each category. So the rotavirus enzyme immunoassay is 60% sensitive versus PCR, which is actually very high, because PCR is so sensitive. But you can see adenovirus all the way over on the left. There's an enzyme immunoassay specific to the enteric adenoviruses, which are group F, types 40 and 41, and it's very insensitive. So again, when you do a study and you find more rotavirus than adenovirus, are you right about that? When we developed this approach, one of the first things we did is tested archive stools we had from a small birth cohort study, about 140 children in Dhaka, Bangladesh. And the red bars represent diarrheal stools by month of life in infants. In that setting, the blue bars represent non-diarrheal stools. So we were astonished to see you're detecting multiple pathogens, 3 or 4 pathogens on average, and maybe you see 4 in a diarrheal stool and 3 in a non-diarrheal stool. This isn't universal, that the bars all the way to the left are in Virginia. The diarrheal stools were what was thought to be a norovirus outbreak, and the non-diarrheal stools, you mainly detect some genes that are associated with types of E. coli that are actually pretty variably associated with diarrhea even in those settings. My daughter is one of the contributes to that blue bar all the way on the left, which is a family tradition because I was in a figure that my dad made a little while back, so it's very satisfying. You can see it increases a bit over the first year of life, but it's really high even in the first months that you see these enteric pathogens. This raises another question about how you do etiologic studies of diarrhea. You can't just test in diarrhea and if you find something, say that it's the cause of diarrhea, because one, they're present in the absence of diarrhea, and two, you're detecting so many that you have to try to adjudicate between them. This is where we work in Haydom, Tanzania, and northwest Tanzania. Still, in our most recent cohort study, about a third get water from the river and from the same sources as the animals, and many more from unprotected wells. So I mentioned this GEM study, this gold standard study. When we had developed these diagnostics and validated them, we had the opportunity to reanalyze stools from that study. This is a case control study. The reason it's a case control study is, like I said, you can't just cross-sectionally look at diarrhea and identify the etiology. You need to compare to what you would see in age and sex and village-matched children in those settings. So this is a fairly large study. We did this quantitative PCR across more than 5,000 cases and matched controls, and they were in age strata in the first and second, and then older children up to 5 years of age. What the figure that's shown below adds the detail that we have quantitative PCR for these pathogens. Because these assays are so sensitive, there's a big difference between detecting logs and logs and logs of rotavirus and detecting rotavirus in trace amounts of nucleic acid that may be just transiently passing through the gut. So rotavirus is very, very diarrheagenic. You can see it on the bottom there. There are darker and lighter columns, and the darker columns represent the number of detections in episodes of diarrhea, and the lighter bar is in the matched controls. The quantification cycle is a measure of the quantity, and as you move to the right, the quantity increases. So actually, even for rotavirus, in very low quantities, you see it as much in children who are well as in children with diarrhea. But as you move to the right, it's very, very strongly associated. What this allows us to do is to decide how much attribution we should give to any detection of any pathogen at a specific quantity, and gives us a fair amount of precision to understand the etiology of diarrhea. This is what we found. So the salmon bars are the original microbiology had been previously published from this study, and the blue bars are what we found when we reanalyzed. I think it's important to note rotavirus, which has been considered the most common cause of diarrhea in the first several years of life, and remains so even with our reanalysis in infants. You'll see that the estimates didn't really change. They're very, very similar between PCR and enzyme immunoassay. I mentioned it was 60% sensitive. I think it's sufficiently sensitive. So probably those additional detections you get by PCR are not clinically or epidemiologically significant. And that's the nice thing about being able to attribute variably based on the detection. Whereas for other pathogens, Shigella is incredibly striking. In the second year of life, that's about threefold higher than was previously appreciated. Not only that, it depends a lot in which site you look. So in one of the study sites was done by ICDDRB, which is the International Center for Diarrheal Disease Research in Dhaka. I call them the Shigella whisperers. They've been doing research on Shigella for decades, and their sensitivity for culture versus PCR was 70–80%. We did a study in Tanzania when we identified Shigella using PCR as the second leading cause of diarrhea in infants, and we didn't culture Shigella successfully in that study. So the correction factor is between maybe 20% to essentially infinite, depending on where you're doing your study. What we found is we really could close the etiologic gap. So we assume that a lot of diarrhea, especially more severe diarrhea, and this study focused on moderate to severe diarrhea in the first several years of life, is infectious in etiology in these settings. But all of the prior studies have really been about half of the diarrhea you could identify, and we've gotten to really close that gap. So it is in the setting of having these diagnostics that I want to go forth and look at the relationship between enteric pathogens and inflammation in particular. And I'm going to use inflammation more as my marker of EED because that's the mechanism from pathogens to growth that's really been best described and best supported by studies. So diarrhea and growth shortfalls has been a moving target. There are a lot of historical studies that suggest that the more diarrhea you have, the less well you grow. This is a meta-analysis by Will Checkley that was published in 2008, but you can see that the studies that are included there are, from 2008, really no studies done within the decade before that meta-analysis was published. And in general, you see the more diarrhea children have, the less well they grow. But there are contemporary cohorts that really haven't supported that. And I'm going to tell you about two of them and work we've done with our diagnostic platform to try to understand that a bit better. There are a lot of theories about why diarrhea is less associated with growth in contemporary cohorts, and I'll touch on a couple of those. So my cheat sheet across the top, I'm switching from, I talked a little bit about the GEM study, which was a case control study. If you have an outcome, namely diarrhea of interest, case control study is a nice design, but if you want to look at a number of long-term outcomes, including child growth and cognitive development, then you really need cohorts. So I'm going to talk briefly about PROVIDE. This was a birth cohort done by Bill Petrie, who's a colleague at the University of Virginia, a fairly large birth cohort for a single site. They enrolled 700 children and included in our growth analysis for 603 children. This is just follow-up to 1 year. So it's very early, as Mark mentioned, for the association between enteric infections and growth. So recognize that. But this is about 3 episodes of diarrhea per child in the first year of life that we retested, again, using the same PCR platform. We saw no relationship between all-cause diarrhea and length at 12 months, essentially nil. But if you look at etiology of diarrhea, attributing using the same approach that we used in the GEM study, you see bacterial and protozoal diarrhea more strongly associated with viruses. One possibility for this switch between diarrhea being a good marker of enteric pathogens and subsequent growth shortfalls and less a clear marker may be that there's been an etiologic shift and there's more, a larger proportion of the diarrhea is viral. It may have to do with the treatment of acute diarrhea as ORS has become available and more aggressive treatment of acute diarrhea and zinc, that perhaps that's also shifted that. These are the most common pathogens identified in this study in Bangladesh, certainly in South Asia, even in travelers now. Campylobacter is appreciated as being the leading cause of travelers' diarrhea, which really wasn't previously thought to be the case. Campylobacter and rotavirus, I mentioned adenovirus is something that people didn't appreciate as a leading cause of diarrhea, but clearly is, as well as Shigella. So we took these top etiologies and then broke it down further. Can you see a relationship between specific causes of diarrhea and growth? So there are 2 bars for each pathogen here. We just use 2 outcomes. One outcome is at 12 months, one outcome is at 2 years. The logic for doing that, just to see, you think of the first year as a very critical time. So how persistent is this association, or do they all catch up and it dilutes out and you really need to follow them further and understand exposures? This is adjusted. This is a kind of a public health level inference because it's comparing high versus low attributable burden. If you look for a single infection, a single diarrheal episode due to specific pathogens, you actually see cryptosporidium as having the strongest effect on growth, but cryptosporidium is relatively rare, especially in the first year of life. So this cumulative effect is highest for Campylobacter. Shigella is the other pathogen that starts to come out, though it seems to go away a little bit when you look at 2 years, and Shigella, again, is relatively uncommon in the first year of life. This is a maternal and child health clinic. These are regional clinics that are facilitated by the hospital that we work out of in Haydom. You can't quite see there probably that there's a scale hanging from the tree, which is where they do the weights. They are waiting. This is actually one of the remote clinics that's accessed by plane. So I'm going to talk a little bit about MALI-D to close. MALI-D was funded at the same time that the GEM study was funded by the Gates Foundation due to particularly persistent researchers who said, look, diarrhea is one thing, but what we really care about is these long-term outcomes. And if we just study diarrhea, then we really won't get at that. And convinced the foundation to also fund this multi-site birth cohort studies. This is done in 8 countries in South America, Africa, and Asia. Included in the growth analysis were 7 out of 8 countries. Sadly, and this represents about 1,500 children. They're followed all the way to 2 years of life. And the nice thing here is that we have diarrheal stools, but they also collected monthly stools even in the absence of diarrhea at all of these sites. And we tested all those. So we just finished testing 40,000 odd stools using these TACMAN array cards. So a generous investment of time and effort by many people that boils down to like 2 figures. You know, that's how it goes. You just hope they're good figures. So again, in MALI-D across all of these sites, it rounded to negative 0.00 per episode of diarrhea, the association with growth outcomes at 2 years. This is a height attainment model. We've also done more complicated longitudinal modeling to try to get better causal inference, but the results are actually pretty similar. Again, you see parasitic and bacterial diarrhea more strongly associated with decrements in LAZ, and viral diarrhea not really at all. So very consistent. There are a couple pathogens that are on this list that you didn't see before. That's because those are pathogens that we don't really see associated with diarrhea in these settings. This includes enteric, E. coli, and Giardia. So they've been added in there, and they're very high prevalence. So they could be very important. So again, to just give you some results, to let some results wash over you. These are separated by bacteria, viral, and protozoal pathogens. And there are 2 bars, again, for each pathogen. Here, we're trying to tease out the kind of pound for pound association between a pathogen and growth at 2 years, and then adjusted for prevalence based on high versus low burden. So the red point estimate shows Shigella to be most strongly associated with growth deficits, but again, Shigella being a little less common. Campylobacter, enteric, E. coli, and you can see Giardia over on the right have a more of a cumulative effect on child growth. We're using longitudinal modeling, something called the parametric G formula, to try to get better, ability to predict the impact of interventions and reduction in these pathogens. And one thing I will say is it's not additive. Again, there's a kind of a certain amount of growth that can be associated with enteropathogens. So I mentioned that these are the inflammatory enteropathogens. Shigella, Campylobacter, enteric, E. coli are all thought to be capable of causing colitis, neutrophil activation. Shigella and Campylobacter are associated with dysentery. We have a marker of intestinal inflammation in the study called myeloperoxidase. And so we looked in the non-diarrheal stools at the association with inflammation in that stool based on pathogen detection. You can see very clearly Shigella and Campylobacter. Plesiomonas is called Plesiomonas shigelloides. It's a Shigella-like organism that causes dysentery. It's relatively uncommon, so you see very poor precision with the estimate. And then enteric, E. coli. So those 3 bacterial enteropathogens most strongly associated with growth are the 3 that are most strongly associated with inflammation. And not just local inflammation, but systemic inflammation too. This is previously published actually using the enzyme immunoassay for Campylobacter, but focusing on the right, in every site, if you had higher systemic inflammation measured by AGP, which is actually mainly used to adjust anemia estimates in the study, but it's a marker of systemic inflammation, higher systemic inflammation associated with worse growth. So I'm just going to finish up very briefly with something else that's related to this topic. So I mentioned that Campylobacter and Shigella cause dysentery, and they're thought of as agents of what's called bacillary dysentery, bacterial dysentery. But it turns out that they often cause watery diarrhea as well, and this becomes very important. So what's shown here is the attributable incidence of Shigella, Campylobacter, and other pathogens for blood in reddish and watery diarrhea in blue. And even for Shigella, about 4 out of every 5 attributable episodes are watery diarrhea. Well, why is that important? Well, there are guidelines from the WHO, the Integrated Management of Childhood Illness, that state that you should really only use antibiotics if a child has fever and blood in their stool, because it's the only thing that's specific enough to suggest that Shigella is present. This thinking has started to change a little bit, or really pushed by Anita Zaidi at the Gates Foundation. Her question about whether we're giving good guidance or we're under-treating childhood diarrhea, which there's a lot of subclinical enteropathogen exposure, but diarrhea still represents a sentinel event. So they are doing a very large mortality endpoint, randomized controlled trial of azithromycin versus placebo, and we've been asked to use our diagnostics in that study to, again, try to identify whether there are subgroups that clearly benefit from antibiotics, and I think Shigella and Campylobacter are high on that list. So last slide, I talked about in new interventional studies, what can we learn from these quantitative PCR assays? Mark mentioned WASH benefits and SHINE, very comprehensive studies done trying to reduce exposure to enteropathogens in the environment. we need to understand why those studies didn't have an impact on growth. And I think looking to see whether enteropathogen carriage was reduced or not will be a really important step in understanding that. I mentioned the diagnosis and management of diarrhea and trying to understand better whether there are subsets of diarrhea beyond bloody diarrhea that should be treated with antibiotics. There are studies to do treatment of subclinical enteropathogen burden. I saw there was a question raised online about giving antibiotics, and azithromycin is the one that people have really focused on. There's studies that suggest a mortality benefit to giving azithromycin in the absence of overt diarrhea, and it kind of goes on from there. So I will stop there, and these are huge collaborative studies with many people. Eric Halpe is my mentor and colleague at the University of Virginia. Gialu developed the tag cards. Thank you.
We have a couple of minutes for questions.
I have a question for James on Wondwossen combined. You talked about azithromycin as one way to reduce bacterial carriage. Wondwossen showed very surprisingly that azithromycin resistance is very high in East Africa, whereas ciprofloxacin resistance is low, which completely refers to what you see in the US. So any speculation on what causes that, and any speculation on what the impact might be for, say, those microbial trials?
Thank you, Ali. So macrolide resistance, definitely macrolide is, one of the leading or most important one when you have Campylobacter or other invasive in children. So yes, that is really worrisome, no doubt about that. But the speculation, again, unfortunately, we do not have the antibiotic use data, considering most of these are extensive small farms or pastoral inhabitants, and we don't have any much for residual use in antibiotics, by the way. We should not assume there is a residual use. Antibiotics at different levels, but it's over. We also do not know whether it is related to direct use, but also remember in a smallholders, the interaction, human-animal direct interaction is a lot. It's a human use could also, from the selected resistant bacteria, already can get to the reverse zoonosis, which is fairly common phenomenon. But we don't have solid scientific answer, but it's more of a speculation. Girish Kanad, if you know of any information.
I think it's a really good observation. I would only add, at least in our site in Tanzania, and South Africa is the other Malady Africa site, there's essentially no macrolide use in children. So I do wonder about that, and I think it may be very regional in terms of macrolide use outside of humans. I wonder. Can I ask one last question? We see in Malady Campylobacter detection as much as half of children at some sites by 6 months of life have already had Campylobacter detected in their stool. Campylobacter in our site in Tanzania, it's about 40% by 6 months of life have had a positive test already. They're essentially spending most of their time on the breast. They're in a wrap a lot of the time. They're not on the floor that much yet, I would think, but it's already so high. The one thing we see is that there's very early cessation of exclusive breastfeeding for the women to go into the field, and the grandmothers give animal milk. Is it possible that more of the Campylobacter exposure is from animal milk than we appreciate?
Thank you. That's an excellent question. Very important. Those questions are very significant. Let's go back a little bit first to the macrolide issue. It's remember even the salmonella example I showed you. For specific antibiotics, the resistance we see often, not always, but of course the direct antibiotic and the resistance as a response is associated, but often there is very commonly. It may not be related to that antibiotic. But again, the genetic co-selection is a very important phenomenon. In both cases, not only Salmonella and Campylobacter, but even in other organisms. Not only that, even as I told you, it may even not be antibiotics. It could be a microbe, co-selected for together antibiotics. So that's important to consider. The second point is very, very important. Could it be from the animal milk? Absolutely, it could be a very good possibility. But you also reminded me from our work in North Carolina, in pigs, okay, not really, but you consider it as a very loosely as animal model. We see about, again, half of piglets at very early age Campylobacter positive. And the sow milk itself also could be, that is what at the end, in fact, Steve Tucker, my former PhD student is currently at NC State, showed quite a bit. There's a number of sequence types we see highly closely related within farms and across sow farm, nursery farm, finishing farm. You see that linking between the sow's piglets or even along the chain. So I think there's a very good possibility that would be some interesting.
To add to that, James, because we talked about that yesterday, I asked him the question how it is in Ethiopia, and he said, yes, young children do get cows when accustomed to another food, it's always boiled. So the mothers seem to be aware of the risks. Raw milk consumption is common among adults, but for the children, typically would be boiled.
Good morning. I had a question that relates to azithromycin and then back to the analogy of the three-legged stool that was discussed this morning. And I'm concerned that we're missing part of the physiology of gut health, and that is the motility aspect. And so in the US, there's something called gastroparesis, which is abnormal gut motility, and we think of it being post-viral. And many of the studies we're talking about today are sort of missing some of the history and physiology of actually the gut motility. And I'm just wondering if there's a movement to include some of those questions. And I actually don't know how you would diagnose it, because in the US we use something called manometry to measure it. And that's important because we're increasingly using erythromycin to stimulate gut motility in the US, and azithromycin is a related drug. And I just wonder if azithromycin is doing something stimulatory that's benefiting children that doesn't have to do with the microbes as much.
I'm sure you want to answer that question, but I'll answer it too. I don't have any data about that. No, sure you have an answer. Well, I'll answer it by way of this. There's a syndrome, irritable bowel syndrome, that was for a long time thought to be primarily functional, not to have an organic cause. But there's been a lot of work. I think Mark Pimentel at UCLA is a gastroenterologist who's done a lot of that work, suggesting that even in the US, a lot of irritable bowel syndrome and diarrhea, predominant irritable bowel, is post-infectious. There's a syndrome of post-infectious irritable bowel syndrome in travelers. Travel and get Campylobacter, and you tell me how long it takes you to get back to normal. And that is, he has suggested, and there's some evidence that you get autoantibodies against glial cells in the gut that decrease motility as part of that syndrome. And that's why you get small intestinal bacterial overgrowth. And that's why in his trial, it was published in the New England Journal, of giving rifaximin to treat SIBO. This is kind of part of that logic. So it may be that with Campylobacter and Shigella in children in developing countries, that gut motility is part of the story. And Jeff Donowitz, who's a colleague at the University of Virginia, has been doing work on SIBO in the setting of EED, and they have a study in Pakistan where they're doing biopsies to kind of have gold standard EED diagnosis and doing breath tests for SIBO. So I haven't heard of anyone trying to do motility studies specifically, but I think he thinks of gut motility as part of the pathogenesis of SIBO. So it is, from that perspective, being looked at. Azithromycin is also an anti-inflammatory. So I think it is hard when you give azithromycin to know which of those pathways is leading to benefit.
Well, that's a great comment and a good idea. I don't know of any data about gut motility in populations that we've been studying. You can get, with looking at some of those sugar tests, you can get some ideas about that. And so that might be something we'll keep in mind as we look at that. Azithromycin,
it's pretty amazing. The Mordor study, 200 and some thousand kids. We get 4 doses over 2 years, and this reduces their mortality. Because that the effect of that drug is gone in at most few days. And then they sit there being exposed most of their life, just like that. So I don't. It's just been very, very curious and very interesting to me. Is there some effect that we need to periodically clean out the dirty closet? Are we collecting microbes in our gut that are maybe some of the bad ones just pile up, and that has an effect with this periodic antibiotics. So antibiotic stuff works in pregnant women to do all kinds of child health benefits, and what's up with that? So, yeah, or is it something that we're gonna find, in 5 years that these were some pretty exciting results, but they're not, sort of changing child health worldwide forever.
I would recommend a Dr. Fuentes of the Tufts in Boston, who's, in my mind, the authority on fertility.
I think at this point, we need to take a break.